Welcome to Spotlights. My name is Reinier and I'm the preLights community manager. And in this episode, we'll be talking to Guy Teichman from the Oded Rechavi lab, who just published a really interesting, exciting new preprint. And we're here to talk about that. So welcome, Guy. Could you tell us a bit about yourself, please?
Thank you. I'm Guy. I'm a computational biologist. I just finished my PhD at Oded's lab a couple of months ago and transitioned to working on genome editing therapeutics.
Awesome. So regarding the preprints, could you, in one sentence, more or less, describe the topic of your preprint?
So in one sentence, in this preprint, we show that the neuronal perception of the environment on its own is sufficient to drive meaningful and heritable epigenetic changes.
Wow. And so to put this in more simple terms, perhaps, what kind of problem does your research try to solve and why is this important?
So our lab studies the field of epigenetics. So how the environment can shape our genetic programs and how those changes can possibly affect future generations. And one of the things we've been trying to tackle is sort of the connection between sensation of the environment and what is the current environment looks like, and the driving of epigenetic responses in CLNs. So in particular, we tried to address the interesting question that not a lot of people tried to address before, whether the neuronal perception of the environment on its own, regardless of the actual change, is enough to drive those changes in epigenetic information. It can change gene expression programs, affect development, and perhaps even affect progeny later down the line.
And could you tell us a bit about, actually doing the study? What was the most challenging bit, for example?
So I think one of the most challenging aspects of this study was trying to develop the system where we were going to probe. Basically, we needed to modify the perception of the environment. We focused on temperature, so we had to basically make the worms think that they're in a different temperature than what they actually are, and do that in the most impactful way, but also in a very specific way that's not going to have other side effects, so that we can get as clean an answer as possible to this question. And it wasn't an easy challenge. We ended up trying using 3 different methods to manipulate temperature substantially in the worm. None of them is perfect, but all of them together, in my opinion, at least covers a lot of ground. They sort of cover each other's weaknesses.
Is this the thing that makes you most proud thinking about this preprint, that it developed these methods, or is there something else?
So there's actually something else that I'm very proud of. So when we worked on this paper, we discovered a lot of phenomena that are related to the effects of temperature sensation on small RNAs and on epigenetic information. Many of those effects were very complex and hard to explain, like in a simple sentence. Like it would be hard to tell you what the phenotype is because the phenotype depends on how long the worm spent in one temperature and which direction they transition to and which neurons you affect in the pathway. And so we weren't, for a very long time, we weren't really sure about what is exactly happening. But near somewhere around the last couple of months, we managed to, after lots of attempts, to develop a mathematical model that proposes a very simple change in the regulation of epigenetic factors that can explain all of the complex phenomena that we observed. And I think the coolest thing is that we managed to make predictions with this model and then verify them experimentally. So that really supported our conclusion and was like going from an idea to suddenly we're able to explain these really complex and unintuitive phenomena using very simple explanation.
Yeah, really cool. And so maybe could you tell us a bit about the questions that naturally flow from this work? What are the things that you can follow up with?
So I think our research opened a lot of questions about how exactly the nervous system can affect the rest of the body cell non-autonomously, not just through activity in the nervous system, but cell non-autonomous signaling. And we do show some pathways that we think are involved in this signaling pathway, but I think it is still an open question to what extent the neuronal perception of the environment can affect epigenetic information and how that information spreads around the body. What are the mechanisms? What are the limitations? And I think it might even be interesting to take this from perception itself to even the processing of information, maybe from just perceiving the environment to making predictions, making the calculations, and
lots of things then.
There might be there's lots of unanswered questions, I think.
Wonderful. Yeah, great. I mean, every good study has that, I guess. Just wondering, so it's always a tricky question, but thinking of the key take-home message, is there one message or several, perhaps?
It's a good question. So I think probably the most interesting finding of the study is just the ability of the nervous system itself to affect other systems in the body, and maybe even do so, at least in worms, in a heritable manner. I think that was very interesting. Another one, which I think maybe is less novel, but was something that I took with me home, is how one simple change in a system can cause like a cascading response that leads to a very complex phenomenon. And maybe that's a bit philosophical, but I think it's interesting in biology how one simple change can drive lots of different phenotypes in lots of different situations.
Well, thank you very much for sharing. And if you want to learn more about Guy's work, please go and check out his bioRxiv preprint. The link will also be in the description. Thank you very much for listening, and till next time.